Hi, my name is CJ and I'm working with an environmental toxicologist in the Fisher lab at UCSF. So there are thousands of industrial chemicals in use today, and less than 1% of these chemicals have ever been tested for toxicological effects they may have on the developing brain. Furthermore, if these chemicals are tested, they're tested on animals, a practice which is undesirable for a variety of reasons. So we're working on developing stem cell-based testing methods in hopes of producing a superior alternative to toxicological animal testing. When this internship is complete, I would like to continue working in the field of neuroscience.
Hi, my name is Jessalyn LaBelle. I'm an intern at UCSF Mission Bay in San Francisco. I work with mouse stem cell models to look at chromosome inactivation. Hopefully, one day we'll be able to understand chromosome inactivation enough to voluntarily turn off chromosomes that we don't want on, such as the third 21st chromosome that's responsible for Down syndrome. After the internship is over, I'm hoping to find a part-time job in a lab while I finish my undergraduate degree in mathematics.
Hi, my name is Ventura Mendoza, and I am a CIRM stem cell intern at UCSF and Blood Systems Research Institute. Currently, we are looking at maternal immune suppression during pregnancy because there appears to be a sort of immunological no man's land in the membrane surrounding the fetus. We hope to determine what sort of mechanisms are in play that seem to turn off the attack signals in the maternal immune cells. We would like to use this information on transplant patients to increase the successful rate of engraftment by downregulating the host immune response. And after this internship, I would like to apply to grad school in either developmental biology or immunology.
Hi, my name is Ekaterina. I'm an intern at the Bruce Conklin Laboratory at the Gladstone Institute of Cardiovascular Disease in UCSF. Heart disease is the #1 cause of death in the world. In our lab, we try to understand the genetic basis of why heart cells become sick. To study cardiac disease, we use healthy human heart cells and then turn off particular genes that are responsible for a heart functioning properly. By modeling heart disease, we can understand its nature and then design drugs to target this disease. In this way, we can work towards new therapies and find better cures. By the end of my internship, I would like to find a position in a lab with similar research interests, and then in the future apply to a graduate school.
Hi, my name is Elizabeth Yee. I work with Dr. Marcus Munch at the Blood Systems Research Institute, where we are part of the Stem Cell Core. My project deals with looking at fetal membranes, the special type of tissue that surrounds and protects a developing fetus. So a developing fetus can be thought of as a foreign body, but instead of the mother attacking this foreign body, the mother's body actually integrates and nourishes it. And we think that this is partly due to the properties of fetal membranes. So I'm looking to analyze and apply these properties in a transplantation setting where we can help donor cells survive in a host. After this internship, I'm looking for a full-time position in research, and I hope to eventually apply to grad school studying developmental biology.
My name is Mike Saxton. I'm an intern in Bruce Conklin's lab at the Gladstone Institute. Currently, I'm studying the use of stem cells to create models that can be used to study heart disease. Heart disease is responsible for roughly 1 in 4 deaths in the US. Unfortunately, heart disease is extremely difficult to study because heart cells, unlike other cell types, do not multiply in a petri dish. This has meant that in the past, we've had to use human subjects to study new drug treatments, which is extremely dangerous, or animal subjects, which is unreliable because their hearts are different than human hearts. Now, using stem cells, we can create heart cells in a petri dish and test thousands of possible new drug treatments in the time we've traditionally only been able to test a few. When this internship is over, my goal is to find a research position in the biotechnology field.
Hi, my name is Earthlight Lowe, and did you know that colorectal cancer is the 2nd leading cause of cancer-related deaths in America? Over the past 20 years, colorectal cancer has been on the decline due to early detection and removal of precancerous outgrowths. For those who succumb to cancer, there are 4 different treatment options: surgery, chemotherapy, radiation, and the newest is target therapy. At Oncomed Pharmaceuticals, we believe that if you can identify that original cancerous cell and block it or remove it using target therapy, you can stop cancer from growing and spreading. My job at Oncomed Pharmaceuticals has been to help identify those potentially hazardous cells and to evaluate the influence target therapy has on colorectal cancer cells. Upon completion of my CIRM internship, I plan to follow my interest and find a full-time position in stem cell research or cancer research here in the Bay Area. Thank you.
Hi, my name is Nick Mendoza, and I'm an intern at the Weiss Lab at UCSF Parnassus, where we study the genetics of autism. 1 in every 68 children in the US is affected with autism, so it's pretty much everywhere. And for every female, there's about 5 males affected. Autism is characterized by repetitive behavior, social impairment, and a deficit in language. So what we do at the Weiss Lab is take cells from people with autism, turn those cells into stem cells, and then we can take those cells and turn them into brain cells and compare them to people without autism. The overall goal is to increase understanding, diagnostics, and treatment. After this internship is over, I hope to get a job in a lab while I finish my undergraduate degree in synthetic biology.